صباح الخير. شو رزان، في صوت الآن؟
موجود هلا دكتور.
تمام، كان في مشكلة بالمايك. طيب خلينا نعمل بس التليفون سايلنت. Okay, today we are going to talk about something already mentioned before and discussed briefly. اللي هو بيتكلم عن signal transduction. And let me share the screen first. Here we go. Here is the laser. Okay. هلا المرة الماضية كنا تكلمنا عن بعض الميتابوليك pathways and how we synthesize certain hormones and the differences between these hormones and why they are very diverse. Why we have these many types of hormones, because we have different actions and a different specificity in the target tissues. So that's why we need to synthesize different types of hormones, even from the point of the component of that hormone, the structure of that hormone. Sometimes it could be just metabolized of certain amino acids, and some others could be fragmented peptides, and some other hormones are synthesized from steroid structures or cholesterol molecule. So we have wide range of origins of these hormones, and that's why we have different actions of these hormones. And today we are going to continue this story of strange action of these hormones by discussing the hormone action and the signal transduction. And you'll find details about this on chapter 42 of the 13th edition, actually, of the Harvard's Biochemistry. So you are very familiar, I think, with the term of signal transduction. It means that how we transfer the signal from the effector molecule to or inside to the cell. So basically, the transduction of the signal or stimulation by certain signal could affect the organism or organ or even certain types of cells in that organism, and it was considered as a challenge or threat to the cells, sometimes depending on the organism. What does that mean? For example, in the unicellular organisms, إذا نتكلم عن الكائنات وحيدة الخلية, their lives actually rely on they respond to the signal, like the light or the heat or certain toxic material. So sometimes they have to respond in order to survive. And after the development of these, due to the developmental differences between organisms, we have different responses depending on the organism itself, or the organ in that organism, or the cells inside that organ. So today we are going to differentiate between the two major types of hormones اللي هم الستيمولاي احنا بنسميهم. And in order to have stimulus, we have to have hormone or ligand molecule or stimulating molecule. So as we learned before, we have two types of hormones or ligands اللي هم لهم خصائص مختلفة بناء على solubility. So the first one needs certain type of receptor, which represents the lipophilic hormones or the water insoluble hormones. They need certain transporters, as well as they need certain receptors in order to have their action, while other hormones just bind a receptor and the signal is going to be transduced inside the cell. So this group is representing the hormones that are water soluble molecules or water soluble ligands, actually, that bind receptors on the surface of the cell, while the other group, group 1, هم الهرمونز اللي they cannot be water soluble molecules, or they are hydrophobic hormones or structural molecules. So they need to bind certain receptor either outside of the cell and then enter the cell or inside the cell in the cytoplasm and will be translocated to the nucleus in order to have the action. So we have subtypes of these hormones اللي هم they bind receptors and they go to the nucleus in order to perform the action, and this is called gene regulation process, or they will affect the gene expression by either activating or stimulating the gene expression or sometimes inhibiting the gene expression. So the action depends on the type of the hormone and how it's going to regulate the process. في بعض الهرمونات ممكن تعمل stimulation للسيل أو للgene expression أو للgene transcription story, and some other hormones are responsible for the inhibition of certain genes in order to regulate other genes. يعني بيكون عندنا جين وسطي اللي احنا بنعمل له عملية inhibition or activation in order to regulate other genes. It depends on the situation. So we have to discuss certain specific examples in order to understand the mechanism or the exact mechanism of the gene regulation by such kind of hormone stimulation. And I think it's very easy when we discuss these details. On the other hand, we have transporters that can be, for example, this is just an example of the mechanisms that can be controlled by such kind of hormones. The hormone will bind the receptor inside the cell and it will regulate the target protein without affecting the gene transcription. So it depends on the situation of that hormone. While the group 2 has diverse effect much more than the group 1 of hormones. Because they bind receptors on the surface of the cell, and gods know what are the signals that will be generated inside the cell as a matter of signal transduction. بمعنى هو راح يرتبط على الريسبتور على السيرفس، وديبندing على هذا الهرمون والريسبتور تبعه we will see many effects inside the cell. Why is that? Because we will have if you would, إنجاز التعبير, sub-signaling molecules or second signaling molecules inside the cell. And it will be like cascade reactions inside the cell in order to have the response. So we could see, for example, certain modifications of certain proteins, translocations of others, and affecting certain transporter channels, for example, or could be other transporters rather than being channels, or it could affect gene transcription. هلا السؤال بدل لذهن البعض انه يعني كيف هذول بيتداخلوا مع بعض؟ الامور لا تؤخذ على مأخذ العزل تماما، انه هذا متخصص في هاي الباثوي وهذا متخصص في ذيك الباثوي. No, we have very complicated integration between the responses inside the cell. So sometimes you will see stimulation of certain pathway. At the same time, the hormone will cause eventually inhibition of other pathway or feedback inhibition in order to have balanced regulation or balanced action. So it's not just white and black. So we have integration between the signaling pathways inside the cell. واللي دخل على الخط جديد molecules اسمهم المايكرو ار ان اي molecules وسمول نيوكلير ار ان اي molecules. هذول لهم كمان دور في regulation inside the cell. So the regulation or signaling inside the cell is very complicated process. عشان هيك عملية الدراسة بتكون أحيانا فقط لبروتين واحد لريسبونس واحد. You could study, for example, just this mechanism, انك تجيب الهرمون and you will see the action of that hormone on just one protein, how it's going to be modified. هلا في بعض الدراسات لا، انت بتروح تتخصص اكثر، بتشوف وين الموديفيكيشن بصير. يعني for example اذا كان هذا الموديفيكيشن للبروتين is phosphorylation. Okay, so the phosphorylation picks up certain amino acids like tyrosine. So the amino acid that's going to be phosphorylated is another story. It means that you can phosphorylate like amino acid number 185 or 93. So the location of the phosphorylation is another story, and you can get a PhD degree in just this response or this pathway. So in fact, scientists still studying these pathways, and they are still find new results or new signaling mechanisms inside the cells. فما في عندنا شيء يعني like the Bible book إنه ABC. We have standards, but we have also interactions inside the cell. So how the signal is going to be generated, as I mentioned earlier, we have ligand that will bind the receptor. So we have ligand receptor complex, especially when we talk about group 1 hormone. بمعنى آخر هذا النوع من السيجنالينج اللي هو جروب 1 لازم يكون الريسبتور مع الهرمون حتى يصير في عندي اكشن. اوكي، بينما في الحالة الثانية الهرمون مرتبط بالريسبتور and then we will have second messenger. هلا مجرد ما تكون السكند ماسنجر ممكن يكون الهرمون لسه موجود، ممكن يكون يعني ما يكون موجود، وبالتالي الاكشن already taken and we have downstream بسموه signaling molecules that will take the action. So it depends. بس بالحالة اللي بنحكي عنها الآن اللي هو lipophilic hormone or hormones, they have to bind the receptor in order to continue the action. So they bind intracellular receptors because they are hydrophobic and they can pass the cell membrane in order to bind that receptor. And then most of the time, these kinds of intracellular receptors hormone complex will bind certain genes and just represent less than 1% of these genes اللي هم can be regulated, I mean. And then we will have kind of transcription, translation, and maybe vice versa. It could be inhibition of transcription and then inhibition of translation. So it depends on the situation of that receptor and hormone. شو مطلوب من الاكشن من هذاك الهرمون. الان زي ما حكيت قبل شويه انه دخل على الخط بالنسبه للريجوليشن جروب اوف ريجولاتوري nucleotides if you would or oligonucleotides اللي هم الار ان اي molecules اللي هم small fragments. كثير بنسمع بالفتره الحاليه عن موضوع الكريسبرز and how they are playing a role in the regulation. المايكرو ار ان اي something like the CRISPR molecules اللي هم regulatory molecules. They bind certain genes in order to activate or inhibit these genes. هلا الكريسبرز في اكثر من نوع للميكانيزم للكريسبرز، بس اشهر نوع اللي هو الكاس 9 اللي بيعمل كليفج للار ان اي او للدي ان اي depending on the binding between RNA and DNA and inhibition of the gene. والان في بومينج وفي يعني ترويج يعني اعلامي كبير بالنسبه للكريسبرز and how can we use CRISPRs in the treatments of certain disorders. طبعا هذا بينطبق على الحالات اللي فيها overexpression of certain genes, so you can target these genes in order to inhibit the action. But still there are huge limitations regarding the use of the CRISPRs, and one of these major limitations is the ethical issue. Are we allowed to make intervention with the human genome in order to treat or not? So there is a debate about using CRISPRs as well as the efficiency or efficacy of the CRISPR's use. But it's a promising technique like the stem cell treatment. Still there is a debate. هلا المايكرو ار ان اي زي ما حكيت قبل شوي هم سمول فراجمنتس من الار ان اي يتكاد طولهم يصل الى طبعا هم بالافرج 20 نيوكليوتايد يعني 20 بيز plus minus. They target complementary sequence in order to either activate or inhibit. Most of the time they are responsible for the inhibition of certain genes. هلا شو علاقتهم بالهرمونز؟ Because they are regulatory, so they are involved in the hormone action in certain ways. Like if you have a signal transduction and then you will have signaling or generation of these microRNA. بمعنى آخر إذا الهرمون عمل signal بعد ارتباطه بالخلية أو دخوله للخلية, you will see generation of these microRNA molecules or small nuclei بسمهم snRNA molecules. So these regulatory RNA molecules have been seen while we have hormonal action. And going back to the simple principle in order to illustrate how these hormones going to act. For example, we have these two examples of lipophilic hormones, اللي هو thyroid hormone, for example, or retinoic acid hormone, as well as the glucocorticoid hormone, how they act. So let's compare these hormones, how they act or how they perform their action or signaling. So first of all, we have such kind of receptor, and this receptor can bind certain element or certain DNA element. هلا احنا مسمينها هون response element، ولكن حرف التي هون just to mention that this receptor or this element is going to be affected by their thyroid hormone. So it means that we have thyroid response element. It means انه ال DNA اللي هون او هذا الجين will respond to the action of the thyroid hormone, but the condition is to have binding between the hormone and the receptor. So if we don't have the hormone, we have such kind of inhibitor. Just a simple illustration, it will inhibit the action of the receptor when it binds the DNA gene or the gene or the target gene. وبالتالي فيش عندي action هون whether if it's going to be activation or inhibition of the gene. بمعنى آخر إن الجين الآن في حالة silent, not affected by this receptor because this receptor is conjugated with such kind of inhibitor. عامل block ما بيشتغل. So after the binding of the ligand molecule per se وحالتنا هي هو الثايرويد هرمون to the receptor, it will cause conformational change on the receptor itself in order to cause dissociation of the inhibitor molecule. So this process is going to activate the receptor itself, but as I mentioned before, we still have the hormone that will activate this receptor, and the action will be taken now, whether if it's going to be expression or downregulation of the gene. Most of the time it's going to stimulate the gene expression. هلا هذا الجين اللي هو الثايرويد ريسبونس element جين، بمعنى آخر إنه الجين موجود هون، ولكن المكان اللي بيرتبط فيه عليه الريسبتور هو اسمه ثايرويد ريسبونس element. هي منطقة في الدي ان اي. كلمة element هي عبارة عن منطقة في الدي ان اي regulatory sequence that will be recognized by this kind of receptor. And as we can see here, it's activated receptor in order to activate the gene expression, and we will have downstream signaling. هلا بعد هيك هذا الجين لما بصير له expression بنشوف احنا الافكت تبع الثايرويد هرمون. بمعنى آخر من ناحية physiology, what you will see is the effect of the thyroid hormone in the cell and how the cell is going to respond. الريسبونس هون كان عبارة عن إنه إحنا بنأثر على الجينز، وببلش الآن هذول الجينز يعملوا transcription translation and expressing certain proteins، وهذول البروتينز اللي هم راح نشوف الأثر تبعهم whether there is a growth or death of these cells depending or metabolic actions that will be taken inside the cell. ففي عندنا actions مرتبطة بهذا الجين اللي صار له activation بسبب وجود هذا الهرمون as a stimulator. هلا لو جينا على stimulator آخر زي الجلوكورتيكويد هرمونز, actually they bind receptors inside the cytoplasm. So the difference here is that the receptors are localized either in the nucleus or, I mean, it's always there, or it's cytoplasmic one. In the case of the cytoplasmic receptor, we can see here another protein which is called HSP اللي هو heat shock protein اختصار، وهم جروب كبير من البروتينات. هذا البروتين is responsible for the inhibition of the receptor itself. Okay, اللي هو المسؤول عن الاكتيفيشن للجين او الانهبيشن للجين حسب الوضع. هلا هذا الريسبتور or خلينا نسميه binding protein that will be recognized by the hormone or the effector molecule is not active. It's not going to be translocated to the nucleus unless it binds the ligand اللي هو الهرمون أو effector molecule. Once it's recognized by this kind of ligand, there will be also a dissociation between the inhibitory protein اللي هو heat shock protein في حالتنا هاي, and we will have the active form. It's very simple principle and it's similar to the previous one, but the difference is this one is cytoplasmic and the other one is nucleus. So after the binding between the ligand and the receptor, there will be translocation, and this translocation is performed or conducted once we have binding between the receptor and the ligand molecule. After the translocation, the receptor will bind the, in this case, of course, glucocorticoid response element اللي صارت G مش T. عشان response element, it means that we have DNA sequence always, and the responding or the affecting molecule will be the cause of the name of that gene. So we are naming the gene or the element depending on the effector molecule. So then we will have activation of the gene, whether we have transcription or inhibition of transcription. It depends on the gene situation. Okay, then we will have expression of the gene most of the cases, and this gene expression will cause certain metabolic action inside the cell, or sometimes it could be growth. It depends on the situation of that cell. احنا ما بنحكي عن التفاصيل هون للشو اللي بيصير بعدين، لانه المطلوب منا الان نعرف البرنسبل اللي هو المولكيولار principle of the action of these hormones. So basically what's going to be affected or regulated by signal transduction انه الهرمون بعد ما مرتبط على ايش بيأثر. We have many levels that will be affected in order to have a response. يعني حتى نشوف الريسبونس بعد وجود هذا الهرمون بعد ما يوصل للتارجت. وين ممكن يشتغل هذا الهرمون؟ It could affect transcription for example, or it could modify the pre-RNA molecule اللي هو immature RNA, so it could be degraded at this level, or there will be degradation of the messenger RNA, so this is second level of regulation. هذول اسمهم levels of gene regulation. How we regulate the gene, it could be at the level of transcription or post-transcription اللي هو degradation of the primary RNA molecule, messenger RNA, or pre-messenger RNA, or you could degrade the messenger RNA by the small molecules of RNA اللي هو micro RNA molecules, and this is another mechanism. Or you could affect the transportation mechanism, and it's very rare to control transportation unless you have certain degradation process before the transportation. And it could be also affected by degradation outside of the nucleus اللي هو degradation in the cytoplasm. And most of the messenger RNA, microRNA molecules or small RNA molecules is going to affect this kind of messenger RNA. لأنه إذا ظل موجود المسنجر RNA, we always have translation. So you have to activate or deactivate this messenger RNA depending on the situation in order to regulate the gene. فممكن تعمل له degradation, ممكن تعمل له inhibition, but it has to be halted. Means that it has to be inhibited in order to stop the action. Okay? Otherwise, you will have always translation and expression of that protein. هلا بعد ما صنع البروتين، طيب بعد انه على سبيل المثال اجى هرمون ارتبط بالريسبتور activation of the gene, and then we cannot control all of these steps. او عملنا كنترول للستبس هذول، ضل في عندي بروتين كمان موجود في الخليه. هذا لابد ايضا انه يصير له ريجوليشن بطريقه التخلص من هذا البروتين by degradation. And in order to degrade this protein, you have to modify it. Modification انك تبلش تعمل عليه نوع من العلامات زي الستيكرز اللي بتحطهم على صفحات الكتاب. So there are kind of proteins اسمهم ubiquitination proteins. هذول بيعملوا tagging للبروتين حسب عمره وحسب نوع الامينو اسيدز الموجودين عنده. ففي عنده مجموعة البروتينات بتتميز بطول عمرها اعتمادا على هذول الامينو اسيدز الموجودين فيهم. كل ما كانوا اكثر كل ما كان عمر البروتين اقصر، لانه هذول الامينو اسيدز يعني بيسببوا له نوع من، خلينا سامحوني على التعبير، يعني بيفضحوه بالخليه انه هي اللي موجود، وبالتالي ببلشوا اليوبيكوتينيشن بروتين يحطوا علامات زي المخالفات انه هذا البروتين is going to be degraded. فكل ما كان الامينو اسيدز اللي بنحكي عنهم اللي هو التاجد امينو اسيدز هذول اكثر، كل ما كان عمره اقصر، لانه اليوبيكوتينيشن بيعمل تاجنج اكثر. ايش يوبيكوتينيشن معناها؟ انه بروتينات بتبلش تعمل certain binding of molecules on these amino acids, and the degradation of proteins will see this protein. في بروتينات مسؤولين عن الديجراديشن للبروتينز اللي عماله بخلص عمرها، اسمهم بروتيوسومز. البروتيوسومز عباره عن كتل بروتينيه موجوده داخل الخليه مسؤوله عن الديجراديشن للبروتينات، اما بتكون ديفكتد او بيكون الهالف لايف تبعتها اقتربت، بمعنى اخر انه الشيلف لايف تبعتها is going to be expired, so they have to be degraded. The point is we have many levels of gene regulation or regulation of the gene expression starts from the gene until we reach the protein. لانه مش معقول انا دائما اضل اعمل جين اكسبريشن، مش معقول دائما اضل اعمل سينثيز لهذا البروتين، وبالتالي من هون اجى دور الاكشن للهرمونز. الهرمونز لازم ترتبط خليني اتخيل انه في عندي السيل ممبرين، انه ارتبط في هذا الريسبتور الموجود على السيل ممبرين، او sometimes the receptors inside the cell in order to affect the gene expression at many levels. فعشان هيك we have to understand in regulation is not just simple story binding with receptor and then we'll have just simple action. It's very complicated action. And when you go deep to study these mechanisms, it's mind-blowing in order to understand these mechanisms. At the same time, it's a matter of joy. You will enjoy reading such things. والحقيقة يعني من الأشياء اللي خلتني أنا كثير أحب البيوكيمستري والمولكيولر بيولوجي is this story اللي هو signal transduction as well as the molecular pathogenesis. The molecular pathogenesis, in order to understand how we have or the body or the organ develop pathological response, you have to understand the molecular biology and the signal transduction. So it's very, very interesting thing to study and to actually go deep and understand the deep details in these mechanisms. Okay, moving to the group 2 of these hormones اللي هم like the peptide and catecholamine hormones اللي هم they are water-soluble hormones and they have receptors on the surface of the cell, for example. Because they have receptors on the surface of the cell, it means that they are going to be in the cytoplasm much more than the other hormones اللي هم group 1. And this will cause shorter half-life. So usually they have short half-life in the plasma because we don't want to have continuous action of these hormones. لأن يظلوا موجودين في البلازما, they will bind the receptor and they will cause the action. And we already discussed one of these mechanisms, one of these hormones, how they work, when we talked about the G protein coupled receptors. وخليني هون رغم انه الحضور انا كثير بيسيئني انه متواضع، خليني بس اشوف العدد قديش اليوم. Where is it? Okay. بس 27 اليوم يا جماعة. طيب anyways متى امتحانكم انتم؟
الخميس القادم كمان أسبوع أو حتى تقريبا كمان أسبوع وشوي.
لأنه إحنا عندنا محاضرة ب2 الشهر. الأسبوع الجاي في محاضرات، تمام؟
ولا الأسبوع القادم محاضرات؟
ولا الأسبوع هذا بس، لأنه أنا التايم تيبل مش أمامي.
لا هو الأسبوع هذا المفروض نختم السيستم.
تمام. لأنه محاضراتي رح تخلص ب2/12. أوكي. almost will be done. أسأل سؤال هدفين، هدف إنه أشوف متى الامتحان فعليا مشان أحضر الأسئلة، الشيء الثاني أشوف إنه الناس حاضرة ولا لا، لأنه أعتقد إنه المعظم بشغل وبروح as usual. طيب هلا going back to the topic itself، زي ما حكينا المرة الماضية أو بالمحاضرات السابقة كنا حكينا عن هذا النوع من الريسبتورز. اوكي، نحكي شوية تفاصيل زيادة الآن، مش رح ندخل في ديتيلز معقدة بالنسبة لهذا النوع من الريسبتور. So I want you to understand the difference between the receptor and the G protein itself. Okay, so this is the receptor that we talk about, and this receptor is called G protein coupled receptor. بمعنى آخر إنه هذا receptor is coupled with this G protein. So this is the G protein that we are talking about. Okay, and this protein consists of alpha, beta, gamma, and you can see here small s on the alpha subunit, which implies there is kind of stimulation. Okay, so this is going to be a gate for the subtypes of the G proteins. In order to make it clear, the G proteins or G protein coupled receptors, their action could be stimulatory action or it could be inhibitory action. Okay, that's why we have to use these small letters اللي هو S or I depending on the type of the G protein. طبعا هذا الشيء بيعتمد على receptor itself as well as the ligand اللي هو الهرمون or the effector molecule. basically the receptor itself. اوكي اللي هو جي بروتين coupled receptor، هيك بصير اسمه كامل لانه ريسبتور هو مرتبط بالجي بروتين. مميز بانه يتكون من 7 helices or 7 helical structures that are integrated in the membrane of the cell. فهم مشهورين هذول الجي بروتين coupled receptors اللي بيعتمدوا على الجي بروتين انهم مكونين من 7 من الهيليكال ستراكشرز، وكلنا بنعرف شو يعني هيليكال ستراكشر اللي هو السكندري ستراكشر اوف ذا بروتين. اوكي and the end terminal is in the outside or outward of the surface of the cell, and the C terminal is at the cytoplasmic direction. Okay, if there is no hormone action, the receptor itself is not going to be changed from the point of view of the conformation. شكله مش رح يتغير, and everything is going to be at the baseline level خلينا نسميه. And the G protein is coupled with the receptor. After that, or if we have certain stimulator like the hormone that will bind the receptor, you can see the conformational change in the hormone structure. الفكرة من الهرمون receptor structure. الفكرة من ارتباط الهرمون أو ارتباط البروتينات بشكل عام مع بعضها بيعود لشيء أساسي بالبيولوجي. لما كنت أدرس بيولوجي للطلاب إنه من أهم الأشياء الأساسية اللي تفهمها عن الماكرومولكيولز إنه they have sense. What does that mean? You know, they can recognize each other. I mean, if they can recognize each other, and they will have sensing mechanism. هذا sensing مش زي ما بنفهمه بالنيرفس system. Sensing معناها إنه you will have certain change in the structure. This is the basic principle of macromolecules. كل الماكرومولكيولز عندهم sensing. وكلمة sensing معناها هنا السنسنج الكيميائي مش السنسنج اللي هو as a nervous system response. على سبيل المثال خلينا نذكر بشكل سريع، في فرق بين الدي جلوكوز والال جلوكوز رايت، وفي فرق بين الالفا اند بيتا جلوكوز، وهذا بيخلي في فروقات بانه بعض الكربوهيدراتس انه can be degraded while others cannot be degraded. Why? Because the enzyme that is responsible for the degradation can sense the molecule. It has very specific recognition site. هاي كلمة sensing معناها. وبالتالي بالنسبة لهذا المولكيول بنلاحظ انه شكله كان هون straight, just a simple demonstration, and we'll have here conformational change that will cause a release of alpha subunit, and you will have here replacement of the GDP with GTP. Okay, so the alpha subunit now has another conformation that will cause dissociation from the gamma and beta subunit or subunit, and it will bind this enzyme. So there is an enzyme which is called adenyl cyclase most of the time that will be activated by the alpha subunit after the GTP binding. This is the basic principle of the stimulation of the receptor that is coupled with the G protein. Okay. Most of the time, the second messenger is going to be the cyclic AMP. What does that mean? After the stimulation here, we will have a generation of second messenger inside the cell. وهذا حكينا عنه يمكن سابقا، يمكن حكى عنه بالفيزيولوجي. I don't know شو الكونتنتس اللي بتاخذوها هناك، بس I'm sure the molecular biology is invading every kind of discipline. So these are the kinds of the hormonal effector molecules that will cause stimulation or generation of cyclic AMP. Just to know the difference between them, انه لو حفظنا هدول بضل البقيه هو عباره عن cyclic AMP activating hormones, but these hormones are stimulatory while others are inhibitory. So the action of the hormone on the G protein, whether it's going to be inhibitory or stimulatory, I mean the alpha subunit, is it going to be S or I, depending on the type of the hormone. So these hormones are responsible for the activation of the I unit or inhibitory unit, or they are inhibitory proteins. And these hormones are responsible for the activation or stimulation of the adenyl cyclase, اللي هو cyclic AMP molecule. So it depends on that hormone. It's not always stimulation of the adenyl cyclase. It can be either inhibitory or stimulatory. But still, we have G protein mechanism. So the G protein coupled receptor means that you have the G protein, and this G protein could be inhibitory or stimulatory to the enzyme itself. Okay. So the action after the generation of the cyclic AMP is by the adenyl cyclase. الأدينيل cyclase هو المسؤول عن إنتاج ال AMP, cyclic AMP. We will have the activation of protein kinases, phosphoprotein formation, and then activation of phosphodiesterase and phosphoprotein phosphatases that are responsible for the dephosphorylation. So we have complicated mechanisms like kinase اللي هو عمليه الفوسفوريشن and phosphatases اللي هي بتشيل الفوسفات اللي هي dephosphorylation. So it depends on the situation that we talk about. هلا these are the examples of the G proteins like S protein or I protein اللي هو two major types. Okay. هلا هذول some other types of G proteins. They depend on other types of enzymes that are going to be activated like الفوسفوليبيز. بينما هون نحن بنحكي عن الادينيل سايكليز لما بنحكي انه هو اس ولا اي، هل هو ادينيل سايكليز راح يصير له activation ولا inhibition. Okay. So and we will have subsequent mechanisms due to the activation of cyclic adenyl cyclase or inhibition of adenyl cyclase. معنى آخر إنه في عندك cyclic AMP أو ما فيش عندك cyclic AMP. While here we have activation of other factors like phospholipases or raw protein that are responsible for other actions, like, for example, in this case, cell shape configuration or cell shape structure. While here we have action on the muscle contraction or blood pressure, for example, due to these kinds of G protein receptors, and that's why they are called GQ, because they don't have adenyl cyclase mechanism. They have other kinds of enzymes that are responsible for the signal transduction later on. Okay, so basically, let's take this example. The stimulus will be glucagon, and adenyl cyclase is going to be activated, and then the glyconeogenesis will be activated by the action of this mechanism. Okay, so lipolysis, for example, glycogenolysis is going to be also affected by the stimulatory mechanism or the G protein coupled receptor as a stimulatory signal. So on the other hand, let's take this example. For example, acetylcholine is responsible for the inhibition of the adenyl cyclase action, and we have slowdown of the heart rate due to such kind of action. So it depends on the type of the stimulator, and the type of the stimulator is determined by the G protein inside the cell, and the action will be due to that kind of G protein, whether it's inhibitory or stimulatory one. لا هون أنا ما بطلب التفاصيل بالنسبة لهذول, just to know the differences, and we have these stimulatory and inhibitory G proteins and GQ proteins اللي هم علاقة ب other types of actions rather than the adenyl cyclase. Why is that? لأنه they have many details inside. Okay. Talking about also the general example here compared to what we talked about earlier. Okay, just give me a second. Okay, sorry for that. Okay, again, if we have certain signal that will affect the receptor and assume that we have we activated the adenyl cyclase, the adenyl cyclase will convert the ATP to cyclic AMP. Okay, the signal has been found in microorganisms, I mean cyclic AMP signaling, that will bind cyclic AMP response or regulatory protein in the prokaryotic cells, and we have certain signaling that is specific to the microorganisms. Assume that we don't want to have the cyclic AMP anymore, so the phosphodiesterase is going to be activated in order to deplete the cyclic AMP. تتذكروا هون كنا حكينا عن الخطوات اللي بتصير بعد الاكتيفيشن through the cyclic AMP second messenger mechanism. So after the formation of the cyclic AMP, it will act by regulating the protein itself. But if we don't want to have this protein or this molecule, it's not protein, by the way, it's a molecule, we have to degrade it. Okay, so phosphodiesterases are many types, and one of them is responsible for the degradation of the cyclic AMP or conversion of cyclic AMP to the 5' AMP molecule اللي هو normal nucleotide. هلا إذا كان الأكشن بشكل طبيعي بيعمل the molecule will bind this kind of receptor اللي هو protein kinase A, and it will convert it from being inactive to be active. And this protein kinase A is a kinase enzyme responsible for phosphorylation. So it will phosphorylate certain proteins in order to be phosphoproteins. And these are the amino acids that are going to be phosphorylated. And you have to know them, like the arginine, lysine, or threonine, or serine. That these amino acids or these sites are very common to be phosphorylated in order to have phosphoprotein. And the phosphorylation requires the presence of ATP as well as magnesium as a co-factor for such kind of kinases. So the C2 here is representing the active site of the enzyme itself. هلا الانزيم اللي هو الكاينيز عباره عن جزء ريجولاتوري وجزء كاتاليتيك. هلا اللي بصير بعد ارتباط السايكليك اي ام بي راح يصير له عمليه ديسوسيشن. الار يونت هذول راح ينفصلوا ويضل السي يونت اللي هم الاكتف اللي بيعملوا عمليه الكاينيز اكتيفيتي. هلا انا في طبيعه الحال انه بعد ممارسه هذا البروتين للاكشن تبعه اللي هو الافكت later on, the protein itself has to be inactivated in many ways. It could be degraded or it could be dephosphorylated by certain phosphatases that are responsible for the dephosphorylation of these hormones or these proteins. هذا الphosphatase could be activated by this molecule in certain unknown way. It depends on the mechanism inside the cell. So you could find another vice versa mechanism that will counterattack the primary action, and it depends on the type of the signaling molecule as well. هلا cyclic AMP واحد من common second messengers, and we have also some similar molecule اللي هو cyclic GMP molecule. Okay, it's also a second messenger intracellular molecule. It's another form of the GTP اللي هو converted by similar enzyme اللي هو الجوانيل cyclase instead of adenine cyclase لأنه الجوانين هو النيوكليوتايد الموجود هون, and you will generate this cyclic AMP molecule from the GTP. Okay, these are the hormones that are responsible for the formation of or stimulation or generation of this kind of second messenger, like atriopeptins, for example, peptides produced in the cardiac atrial tissues that are responsible for certain actions of the blood vessels at the physiological level. Okay, we have also other kinds of stimulators that are responsible for the generation of cyclic GMP as a receptor, as a second messenger, like the nitric oxide, one of the most common stimulator molecules, and it's a gas molecule that are responsible for vasodilation, as you may know. Okay, this kind of mechanism is also inhibited by phosphodiesterase, and there are some kind of drugs that are responsible for the phosphodiesterase activity, and one of them is the Viagra. The mechanism of the Viagra is responsible for the activation of the phosphodiesterase. Okay, moving forward to talk about another type of signaling mechanism that involves calcium or phosphatidylinositol mechanism. It's completely different from the things that we talked about before, and this is because we have different types of hormones or effector molecules. Okay, talking about the calcium molecule as a signaling molecule, we have to know that the concentration of calcium as an ion is between 0.05 to 10 micromolar as a concentration. It's very low, it's present in very low concentration, and the level of the intracellular calcium changed from 0.05 to 10 is a huge change. So there are kind of mechanisms that are responsible for the changing of the calcium concentration as an ion inside the cell, and these mechanisms are classified to be three mechanisms. Certain hormones, by binding the receptor, they are as signaling mechanism. The calcium channels are going to be open because these calcium channels are conjugated with these kinds of receptors. So there will be a receptor that is conjugated with the channel, or it can be itself as a channel that will be affected by the hormone, after the binding of the hormone, there will be activation of the influx of the calcium by the binding of certain hormones. Okay, some other hormones they can act indirectly. الهرمون الاول كان بيهاجم بطريقه مباشره لانه مرتبط بالريسبتور اللي هو عباره عن conjugated with the calcium channel وهو بيعمل كنترول للكالسيوم influx, while in indirect way the hormone will promote calcium influx by modulating the membrane transportation mechanism, and it will cause depolarization or can be caused by depolarization in order to open the gates for the calcium. So in the second mechanism, we have gates that will be opened by the action of the hormone after depolarization of the membrane. And the last mechanism, it depends on the first two mechanisms actually depend on the extracellular calcium, while the third one depends on the intracellular calcium that is already stored in the endoplasmic reticulum. So after certain signal transduction, there will be a release of the calcium from the endoplasmic reticulum to the cytosol in order to have the action as an ion. And this mechanism relies on the presence of a calmodulin. I mean, the calcium is going to act after binding the calmodulin. So releasing of calcium from the endoplasmic reticulum or influx of the calcium, the action of that calcium depends on the binding between the calcium and calmodulin. So the calmodulin is the key molecule in this mechanism, and it's a protein that is localized in the cytoplasm, and the action will be it will be activated after the binding of the calcium. And usually there are 4 ions, or there is a requirement for 4 ions of calcium in order to activate this kind of calmodulin. Okay, and it can be explained by this structure. So this structure is representing the calmodulin structure، وهذا الكرتون structure تبعه إذا بدنا يعني نمثله بشكل بسيط جدا. And these are the sites of the calcium binding. We have 4 sites for the action or for the binding of the calcium in order to have the action. And this calmodulin is now going to be activated, and it will bind certain target proteins in order to activate or inhibit depending on the target protein. So the calmodulin is responsible for the regulation depending on the presence of the calcium. So the calcium also depends on the action of other previous stimulators. Okay. هلا زي ما احنا لاحظين بالرسم هون انه الستركتشر او الفانكشن تبع الكالمودولين is almost structural. It binds something in order to have the action. So the regulation of the calmodulin or the calcium with the calmodulin is going to be structural element or structural changes inside the cell. And one of the most affected molecules اللي هم actin-myosin complex of smooth muscles. So that's why the calcium is responsible for the regulation or the action or the mechanism of the actin-myosin or muscle actions. So various microfilaments mediate process in the non-contractile cells are also regulated by the calmodulin calcium mechanism. Cell motility as well is also controlled by calmodulin mechanism. So this molecule is going to have the action as a structural modification molecule. On the other hand, the calcium itself, rather than binding the calmodulin, is a mediator of hormone action. And as a mediator of hormone action, it means that the calcium is going to affect other pathways rather than the activation of the calmodulin. So how they found this, how they found that the calcium is responsible for or can be a mediator for the hormone action. They realized that, blunted by calcium-free media or when intracellular calcium is depleted, they noticed that there is a change in the action of that hormone. So that's why they proposed actually or concluded that the calcium is a mediator for the hormone action. Can be mimicked by agents that increase cytosolic calcium. For example, if they replace the calcium with certain mimicking agents, that are similar to the calcium, they notice that they will have the same action. I mean, the hormones will have the same action, as well as influence cellular calcium flux after certain actions. I mean, efflux from the endoplasmic reticulum or influx from the cytosol, depending on the uh, type of the triggering molecule. Okay, and then because of that, they found that there are certain metabolic enzymes, and they are very critical, can be regulated by the calcium and the phosphorylation mechanism, or sometimes by both. And one of these enzymes or group of these enzymes include glycogen synthase, pyruvate kinase, pyruvate carboxylase, اللي هم almost housekeeping enzymes that are responsible for the carbohydrate metabolism in our body. They rely on the calcium mechanism in order to be regulated. Okay, and this is just explanation of what we discussed before about the calcium story. So there is an action of the calcium after influx due to the certain signaling mechanisms that will cause influx of the calcium from the from the extracellular components to the cell, and then the calcium is going to perform its action by binding of certain proteins, whether in the mitochondrial, for example, proteins, or calmodulin in the cytoplasm, in order to stimulate or to activate certain mechanisms that are responsible, for example, for the motility or metabolic actions. And here also we have the inositol triphosphate, if you remember, that will be generated from such kind of receptor, which is also G protein coupled receptor, but it's Q here, that will activate phospholipase, and in this case phospholipase C is the enzyme. وبكسر الفوسفوليبيد الموجوده في الممبرين اللي هو الانستول دايفوسفيت or phosphoinstol diphosphate to produce diacylglycerol molecule, for example, and انستول triphosphate. هلا اللي بيهمني بهذا الموضوع انستول triphosphate هو مسؤول عن release of calcium or flux of calcium from the endoplasmic reticulum to the cytosol. So we'll have double action of calcium release. It could be indirectly by the hormone that will bind the receptor and activation of certain mechanism and influx of the calcium, or flux of the calcium from the endoplasmic reticulum, and we will have double action. إنجاز التعبير اسمها double action, but it means that we are or the mechanisms are supporting each other. هلا ما ننسى هون كمان انا شايف في عندي بروتين سي انه بيعمل كاينيز اكتيفيتي، وحاطين عليه نقاط لانه بدوش يذكر التفاصيل، but it's similar to the kinase A that we described before اللي بيسبب السايكليك اي ام بي از سكند ماسنجر، بس بالنسبه لنا هون السكند ماسنجر هو الداي اسيلجليسرول molecule. So this is just to have like brief description and integration between mechanisms زي ما حكينا قبل شوي، انه في integration between the mechanisms of the signal transduction inside the cell. انا اعتقد ان الوقت خطفني هون وتجاوزنا، معلش مضطر اطلع، اذا في اسئله بنناقشها بالفلكسبل ليكتشر. and here stop sharing لانه في محاضره اعتقد بعديه. thank you.